Hello, my name is Kyra.
Hi, I'm Ale.
And as a certified genius of the quantum biology ecosystem, I am going to be explaining the sample stage aspect of the quantum microscope.
I'm planning my version of what this is.
Okay. Okay. Can I touch? I don't want to touch anything. So this is an optical wire cable.
As Kyra said, this is an optical fiber.
This really makes sure that the visual aspects are all connected. Honestly, yeah.
The goal for that is to provide illumination at a very shallow angle in order to get the conditions for what is called turf microscopy.
This center area is where the proteins are going to sit and then be electrified or magnified or hit with laser beams.
We're gonna hit our glass slide at a very shallow angle, and that is gonna excite with a very peculiar type of light our proteins, our cells, and whatever we're imaging, and this is gonna create fluorescence.
I don't know why there's a little triangle. Maybe they just want to be like a little bit angled or like a little slide situation, and I'm unsure, to be honest.
The idea is to couple the light into the optical fiber. The optical fiber is gonna guide the light into here, and it's gonna couple to this other optical fiber that is going into this little triangle here, but it can also go into this triangle over here.
Okay, so then you move downwards, there's like another slide situation. They're gonna put light down the chute.
And we can pick up that fluorescence via an objective that is located at the bottom there. The objective collects the light, and then the light goes down there. There's a 45-degree mirror, and the mirror basically reflects the light this way.
And hit this lens.
Basically, the light is coming out of this tube, and then we have this, which is a flippable mirror, so that we can choose if we're deflecting this light or not.
And then it's gonna hit the thing, the photon capturer.
And we're gonna have the light going through, so when this guy is down, going to our single photon detector for the quantum part of the experiment, and then we can just swap, and basically the light that is collected by the objective will be reflected and go to the camera that is going to detect the biology of our experiment.
Nailed it.